I'm happy to welcome my co-host.
So once again, good morning or good afternoon, dear colleagues. I represent Novosibirsk Federal Scientific Research Center. We work with environmental problems of infections and invasive diseases in the Russian Sea and river areas. Russia is one of the biggest countries which has one of the most water areas, more than 8.6 million square kilometers, which does not include lakes, which is the home for many marine mammals. It doesn't include estuaries, which also have feeding grounds for marine mammals. Water has basically all types of microorganisms, including pathogens, and it is considered that microorganisms and the number of microorganisms in water is up to 10 to the power of 5 cells per milliliter. But due to the microscopic studies, we can see that this number of viruses even more. Of course, it depends on the depth and depends on the distance from the shore. Viruses invade basically different organisms. We do not really know the ways how they get inside the organisms and how they transfer from one animal to another. We don't really know much about reservoirs of these viruses in nature. Some of them are really harmful and dangerous for the health of people. And I have to repeat, and everybody already said that the anthropogenic impact is very harmful to the environment. It produces a lot of toxic wastes, and the poor drainage systems also negatively impacts on the environment and produces a good foundation for these viruses to thrive. So now to the diseases themselves. Most of them are induced by pathogens which exist for a long time in the environment. They circulate among these mammals and they are adjusted pathogens. Some of these pathogens appear in marine mammals. They adapt to the new conditions or they find a new way to get inside the new population. And in some rare cases, there is a new variant of a microorganism which has a new infection like influenza or the COVID-19. Oftentimes, we don't really know a lot about the reservoir, and oftentimes we make mistakes thinking that we know about the final reservoir of this or that pathogen. We have quite a number of long-staying pathogens, Anhydrolutris, Dorseps, Truncatus, Orchinosorca, and so on. I will not go into details. You can screenshot it and then read it later. We have this information for different marine mammals during different years. These pathogens circulate among marine mammals for many, many years. They adapted to them and they do not produce any lethal events. So what does happen? So what happens when conditions change? The main thing is the anthropogenic wastes. Because of the river bringing the waters to the Russian water areas, these rivers goes through anthropogenic industrial areas, through big cities with poor sewage systems. They go through plants, they go through factories, and because the rivers go through these industrial territories, it is important to spread the infectious diseases which should be diagnosed within the marine mammals. This list should include not only pathogens for marine mammals from the previous slide, but also new pathogens for marine mammals human pathogens, agriculture, animals, pathogens, and domestic animals, pathogens. Quite a number of scientific literature provides the background, and it says that the closer it gets to human activity, the more diseases we could have based on the data we have. And we have accumulated this data while working and trying to find pathogens and antibodies to different pathogens in beluga whales in the Sea of Okhotsk, the beluga of Okhotsk, and bottlenose dolphins from the Black Sea. Here we demonstrated that in the Black Sea, the anthropogenic impact is very high. We can see that antibody for bottlenose dolphins is 2.5 up to 10 times more compared to the same cetaceans from the Sea of Okhotsk, where anthropogenic impact is considerably lower, 30 even to 100,000 or 30 to 100 times lower. So animals living in anthropogenically impacted areas have more antibodies, but they suffer severely from these conditions. I also would like to mention that during the last decade, anthropogenic impact has increased when we speak about polar bears. The polar bear is the top of the trophic chains in the Arctic region. Due to climate change, it moves to the south, closer to people, and because of that, there is an increasing risk that they will come in contact with new pathogens, with new diseases, which might transfer from domestic animals and from humans. We extended the indication for these new additional pathogens for the polar bear. We had some data, some samples, but still we were able to indicate these classical pathogens, which are usually taken like Toxoplasma gondii. We also checked alpha herpes virus 1, gamma herpes virus, M. hominis, M. pneumoniae, Aspergillus fumigatus, Opistorchis, Anisakis, Trichinella, and others. Here on this slide, you can see the results of this work. Quite a lot of marine mammals which have antibodies to these pathogens beside 3 specific ones. 3 types of microorganisms: Mycoplasma hominis, Mycoplasma pneumoniae, and Chlamydia trachomatis. All of these are human pathogens. They are transferred respiratory with close contact, and these microorganisms are not stable in the environment. So the animals which we checked did not have enough contact with the human activity, products of the human activity, so they were not able to, so the pathogens were not able to transfer from this human activity from the products of human activity to marine mammals. Another reason is industrial accidents and other accidents, for example, floods. This year we had a lot of snow during this year, and I think we will have a lot of water in spring. And as a result, Arctic systems, seas, estuaries will have a lot of problems to handle during this thawing period. There are a lot of pathogens and eggs of pathogens during this time. The increased risk of these new pathogens coming with water from rivers into seas. After floods, we can see deaths of sensible animals with high level of sensitivity rather. There could be outbursts of epidemic for marine mammals with lethal infections, because we don't really know the sensitivity level of this or that marine level to this specific pathogen. And not always it's all possible to see these beforehand. We can trace pinnipeds, we can trace some other animals. They will go to the shore if they feel sick, but it's much harder to work with cetaceans. During the bad weather, we will be able only to see the decayed carcasses, which will be strangled, or which will be stranded on the shore, but we will not be able to help it while we still could. There was another article dedicated to studying beluga whales, which found increased level of antibodies to these new pathogens compared to the previous studies 3 years ago. Speaking about new pathogens, we don't really understand the natural reservoirs of viruses and how they induce diseases. We don't really know about their capabilities to transfer via land systems. It's really hard to understand how it could be dangerous to marine mammals, how dangerous it could be to humans. One of these new pathogens is influenza virus. Pinnipeds and cetaceans were infected with human influenza. We could see the traces of human influenza strain H4N6 influenza. Bird influenza was also noticed to transfer from birds to marine animals. I believe I'm a little bit out of time. Another menacing phantom is the drainage system. Wastewaters. The new virus goes to the wastewater. There are a lot of publications all over the world which speak about this new virus, SARS-CoV-2. And I would like to speak about this publication, which assessed the mutation of the genome, which connects with the SARS. In this case, here you can see on this slide, the susceptibility to SARS-CoV-2 in orange and red. Red is higher than human. And in most cases, we see that for most marine mammals, most of these marine mammals have higher level than humans. They could be infected with SARS, it could adapt, it could change and be dangerous not only for marine mammals, but also for humans in the future. We don't really know how this evolution goes on, how it will go in the new host, how the pathogen potential would change. Everything is interconnected. Pathogens, wild animals, domestic animals, humans, all that is really interconnected, and we don't really know a lot about marine mammals. We need to have a comprehensive system to learn about the pathogens, new pathogens of marine mammals. Identification of this pathogen is very important work, and that's basically all I wanted to say. Thank you for your attention. Do we have any questions? I'm not sure if we have. No, we don't seem to have any questions. Okay. Thank you, Alexander, for your presentation. Thank you. I think I was very fast, and that's why, and it was a new topic. Maybe that's why there are no questions. What are the signs to marine mammals when they are infected with the new coronavirus, SARS-CoV-2? As far as I can understand, this question refers to this pathogen. The article regarding pathogen for cetaceans, it did not say anything about the signs of new pathogens, but what I can say is, or my opinion is, they should have this lung disease just as humans do. some kind of lung infection. If we spoke about pinnipeds, then we could have some more substantial evidence, because pinnipeds are very similar to humans in this regard. But when we speak about cetaceans, I'm not really sure. Probably they should have some kind of lung infection, some kind of pneumonia, I mean. I hope that this answers your question, but once again, this is my opinion. I'm not really sure about the question which I see in the chat regarding the evaluating exposure to microorganism. Thomas, I'm not really sure about the question who you are asking. Yes, I'm asking you, Alexander. You spoke about viruses during the floods. There is a lot of dead animals, and we have more pathogens during the floods. And now it's very interesting to know that a lot of scientists study environmental DNA. Okay, Thomas, I understand your question now. Yes, it would be a very interesting work to do. Now it's popular to study new coronavirus, wastewaters, different types from farms, from industrial regions, from sewage systems. But we don't really pay attention to the fact that besides coronavirus, there are a lot of other potentially harmful pathogens and microorganisms, including viruses. And yes, I agree that it would be great and to have this kind of work during floods, or maybe just to have some samples during shower rains and during floods, and to analyze both SARS-CoV-2 and other new pathogens. And we have all the necessary tools to do so. Well, thank you for your answer.